The G. Evelyn Hutchinson Award honors a scientist who has made considerable contributions to the aquatic sciences. Their future work promises a continued legacy of scientific excellence. The 2021 Hutchinson Award is presented to Dr. Elena Litchman, a professor of aquatic ecology at Michigan State University in the United States. Dr. Litchman is perhaps best known for her work developing a traits-based approach for the study of plankton ecology. Application of the traits-based approach, originally described in terrestrial plant ecology, to phytoplankton community dynamics has been transformational for the field. Litchman's 2007 and 2008 papers, which were the first to describe the traits-based approach for phytoplankton communities, have collectively been cited more than 1,000 times. Since then, Elena has seamlessly integrated observational, experimental, and theoretical models and approaches to understanding plankton community structure and function across freshwater and marine environments. Litchman continues to apply this framework to answer important questions in phytoplankton ecology, such as impacts of climate change on communities, with a research program that is both highly collaborative and international in nature. In addition to having an impressive body of influential literature, Dr. Litchman is involved in several large-scale projects and initiatives, both within the US and abroad. She sits on several advisory boards, including Denmark's Center for Ocean Life and the European Network of Mesocosm Facilities. Elena has also been honored with the US National Science Foundation Career Award, Presidential Early Career Award for Scientists and Engineers, the Michigan State University Foundation Professorship, and the Peterson Foundation Excellence Professorship Award in Germany. ASLO President Roxanne Moranger notes, displaying both depth and breadth in her scientific endeavors, Dr. Litchman exemplifies a highly impactful mid-career aquatic scientist. Perhaps most impressive has been her ability to cross scientific boundaries, including the fresh-salty divide. Her leading-edge work on using functional trait approaches in aquatic ecosystems has inspired an entire direction of research to better understand the relationship between plankton community structure and major biogeochemical cycles. Please join me in congratulating and welcoming the 2021 G. Evelyn Hutchinson Awardee, Dr. Elena Litchman.
Thank you very much. Thank you, everyone. So I'll start sharing my screen. Thank you, everyone, for tuning in on this Sunday afternoon after almost the whole week of conference talks on Zoom. I very much appreciate it. And I'm thankful to ASLO for awarding me this award. And I'm grateful to my nominators, letter writers, and to all my collaborators and students and postdocs. And so I will try to make my talk not too long because it's one of the last talks. So let me see if I can advance slides. So G. Evelyn Hutchinson was one of the greatest thinkers, ecological thinkers of the 20th century. And probably most of you are familiar with his many contributions to aquatic ecology and ecology in general. So the paradox of the plankton that Hutchinson formulated, it was one of the major foci of my research, starting with my PhD thesis. And then many of us know his fundamental treatise on limnology, which I read that he initially named The Natural History of Lakes. And then another exciting concept that really permeated a lot of ecology, both aquatic and terrestrial, is the ecological niche as an n-dimensional hypervolume concept. And there are many, many others. So I'm really, really humbled by receiving this award. So in my talk today, I will briefly discuss how I got here, basically my path to phytoplankton. Then I will present some example studies from my lab that use traits, and I will show how traits can be useful to explain patterns of phytoplankton community composition and dynamics. Then I will briefly mention some exciting new directions that the community is pursuing, that aquatic researchers are pursuing. And then I will finish with some concluding thoughts. So my path to phytoplankton has not started with phytoplankton. As many other kids, I was fascinated with dinosaurs. And I remember, for some strange reason, when I was a teenager, I thought it would be really, really cool to create a mathematical model to understand why dinosaurs went extinct. So that led me to pursue an undergraduate degree in ecology at Moscow State University, Russia, because ecology seemed the most appropriate field to pursue that question from my teenage years. So I set out to model a steppe ecosystem, so it wasn't dinosaurs. So in particular, I was focusing on modeling locusts. And it was a fascinating project. However, I developed hay fever, and I really couldn't go into the field and collect data for my model. So I decided that I needed to choose a different project. And luckily in the department, there was another modeling project on phytoplankton. So basically, I started modeling phytoplankton communities and doing experiments with them. And I defended my undergraduate thesis on modeling phytoplankton at Moscow State University in Russia. So while I was working on my thesis, I became fascinated with the work of Dave Tillman at the University of Minnesota. And I was really impressed how he combined ecological theory and experiments, and it seemed so elegant and exciting to me. So I applied to do my PhD at University of Minnesota, and I was accepted. However, when I got to Minnesota, I realized that Dave Tillman had stopped working on phytoplankton. So I was kind of a little bit of an orphan there. And luckily, Bob Sterner, who was also a faculty member there, he took me under his wing, and I set out to pursue various questions in phytoplankton ecology. And I remember two pieces of advice that stuck with me while I was a grad student. One was from Dave Tillman, and he always told his graduate students to look for the big picture. And I think that is really important, and that allowed me to answer a lot of different questions throughout my career. So another piece of advice I got from Bob Sterner. So when I joined his lab, everyone in his lab was doing really exciting research on zooplankton, and I was working on phytoplankton. So I thought, maybe I should switch to zooplankton. So I asked Bob whether I should switch to zooplankton, and he said, follow your heart. So I followed my heart, and I stuck with phytoplankton. And I have no regrets, obviously. So a while ago, I discovered this the tree of Hutchinson's intellectual descendants. And so I am humbled and excited to point out to you that I am also a little branchling on this tree. And this tree is in a need of updating. So if you take this little branch, so you can see that Peter was one of the descendants, and he trained Dave Tillman. Dave Tillman trained Sterner, and then they both trained me as their PhD student. So the overarching question in my research is what determines the structure and dynamics of ecological communities? And although my initial sort of familiarity with phytoplankton was accidental, I really think it's serendipitous, because phytoplankton are really an excellent model system in ecology. And it all started with the paradox of the plankton by Hutchinson. Then many of us know the resource competition theory developed for phytoplankton first, and then applied to many other systems. And then ecological stoichiometry was also developed with phytoplankton and zooplankton in mind. So in addition to being an excellent model system, phytoplankton are globally important. And of course, I am preaching to the choir here, saying that, yes, highlighting that phytoplankton contribute about half of global carbon fixation happening on Earth, and they're key players in major biogeochemical cycles. They form the basis of most pelagic food webs, and they have a tremendous effect on water quality, both in freshwater and marine systems. So phytoplankton, I still think, are one of the best systems to study in ecology. So when I had that question in mind, how do we best explain phytoplankton community composition and dynamics? And I was pondering on it. And because I was familiar with research in terrestrial plant ecology, I was excited by all the new developments in that field happening in the early to mid 2000s. And so there were all these excellent papers being published on predicting changes in community composition and ecosystem functioning from plant traits by Sandra Lavorel and Eric Garnier. And so many other papers, they really started this exciting new field where we could use traits to predict and explain patterns in community composition. So if you're curious what a trait is, here I provide a definition, one of the possible definitions. So a trait is any quantifiable characteristic of a phenotype or an organism, which is presumably governed by a specific gene or a set of genes. So it connects genotypes and phenotypes, basically. And so these early studies showed that many traits are correlated with each other, and they're correlated with environmental variables. And so looking at traits, researchers in terrestrial plant ecology were able to really explain the mechanisms structuring terrestrial plant communities. And so it was really exciting to me. And I thought that perhaps we could do that with phytoplankton. So we started with rather simple and straightforward questions. So we wanted to know what the relevant traits were in phytoplankton that we should focus on. And we wanted to know whether major taxonomic groups differ in their traits, and whether different traits are correlated, and whether some of those correlations may represent trade-offs, and then trade-offs could give rise to contrasting ecological strategies. So, and probably one of the most important questions is whether we can use traits to explain community composition and dynamics. And then another question at the intersection of ecology and evolution is whether traits can evolve. And of course, our intuition tells us, yes, of course, traits evolve on multiple time scales. So before I start presenting some examples from our work, I would like to thank all the students, postdocs, and collaborators, and not all of them are represented here. And I want to acknowledge funding that we received over the years. And I wanted to highlight my long-term collaborator, co-conspirator, and husband, Chris Klausmeier. So without his input, I would not be giving this talk here, and I really appreciate his contributions. So to answer the question, what the relevant traits are in phytoplankton, we try to look at traits that people measured before. And so we wanted to classify those traits according to their ecological function and trait type. And so this is an old table from one of our papers. And you can see that there are a lot of gaps here. And since then, we filled those gaps, of course. So, but some of the important traits, and probably many of you, of course, know them. So are cell or body size, cell shape, and then nutrient uptake traits, temperature traits, and I will be mentioning some of those traits further in my talk. So another question that we had is whether major taxonomic groups differ in their traits. And of course, our intuition tells us that they might, but there may be a lot of variation that would obscure those differences. So we started compiling data, existing data on traits, and this effort really took off thanks to our former postdoc, Kyle Edwards. He really collected, I think, one of the best databases of phytoplankton traits. And he set out to analyze those data, and he found a lot of interesting patterns, and he published together, we published a lot of these results. And so some of them are highlighted here. Basically, for both freshwater and marine groups, we see that these major groups differ in their maximum growth rates, in their uptake capabilities for nitrogen, for phosphorus. So, and these differences really, they shape the structure and dynamics of phytoplankton communities. And despite a large variation in among traits of individual taxa, we do find some significant patterns and significant differences across groups. So another question, as I mentioned, that we had is how different traits are correlated, and understanding and identifying those correlations can help us simplify our thinking, because we then do not have to explicitly consider all the traits. We can consider some traits and think about others as correlated with the traits that we're focusing on. So again, so the databases that we collected allowed us to look at many trait correlations. And here are some examples. And those trait correlations, some of them represent trade-offs. So basically, and then those trade-offs can lead to contrasting ecological strategies. So here's an example of a correlation between the optimum irradiance for phytoplankton growth for different species, and alpha is the initial slope of the growth irradiance curve, which basically could be thought of as a proxy for light competitive abilities at low light levels. And so you can see that there is a negative correlation. And if we think that some species occupy different positions on this trade-off curve, we can distinguish different ecological strategies. For example, there are clear differences in traits between the high light adapted species and low light adapted species. So, and we found a lot of these correlations and trade-offs analyzing the trait databases, and that helped us explain various patterns that we're finding. So we don't have to stop just looking at pairwise correlations. We can think about multidimensional trait space. For example, here an example of the relationships between three traits, this alpha, the low light competitive ability proxy, phosphorus competitive ability or phosphorus affinity, and maximum growth rate. And different species, as again, occupy different places in this multi-dimensional trait space. So basically, the traits help us understand these different niches, ecological niches and strategies that different groups of phytoplankton occupy. So as I mentioned, a key question is whether we can use traits to explain community composition and dynamics. And so basically, here's an example where we used three traits: scaled nitrate affinity, growth affinity for light, and maximum growth rate, to explain the temporal dynamics of various phytoplankton in the English Channel. And the relative importance of these three different traits varied with season. And we were astounded to discover that 3 traits measured in the lab could explain up to 80% of variation in phytoplankton abundance of different taxa. So traits really can be powerful predictors. So another question, related question, is whether we can predict, use traits to predict community composition and dynamics under global change. And of course, temperature is one of the major global change stressors. So it makes sense to look at temperature-related traits. So a lot of the ocean is predicted to warm up to 4 or maybe 5 degrees Celsius, depending on the models. So what we did, we focused on these temperature-related traits. And here you have the growth response to temperature of various phytoplankton taxa. It's a schematic. So some of the relevant traits are the optimum temperature for growth, the niche width, the minimum and maximum temperature. So 2 fabulous graduate students, former graduate students, Mridul Thomas and Colin Kramer, set out to explore whether there are patterns in temperature traits and whether we can use these traits to predict community structure in the future. So we've assembled traits from more than 150 species of marine phytoplankton, and since then we added a lot of freshwater species, and we published extensively on that. So what we found that obviously different species have different temperature traits, and when we use those traits to predict the shifts in fundamental thermal niches, we found that a lot of temperate species would move towards the poles in the future, and many tropical species would extend their fundamental thermal niche further, again, towards the poles. So when we put all this information together, we were able to predict future changes in species diversity just due to temperature. And we found that tropical phytoplankton may be especially vulnerable to rising temperatures. And we predicted that just due to rising temperature, phytoplankton may decline in diversity up to 30% in the tropical regions. So that was quite sombering. So when we presented those results, everyone was asking us, well, what about other stressors? And of course, one of the main phytoplankton drivers are nutrients, and nutrient limitation is predicted to increase in the future. So we wanted to know whether nutrient limitation affected temperature traits. And again, we did a series of studies where we showed that various phytoplankton, including this model diatom, Thalassiosira pseudonana, they change their temperature curves depending on nutrient levels. And not only maximum growth rates declined under nutrient limitation, but also temperature optima declined. So basically, under nutrient-limited conditions, phytoplankton may become more sensitive to higher temperatures because of the lower optimum temperatures. So taking that information into account, we could predict, again, the thermal niches in the presence of nutrient limitation. And we found that even tropical species would have regions in the ocean due to nutrient limitation that would be too hot for them to survive. So basically, this nutrient limitation may exacerbate the effects of high temperature. So, well, it's not all doom and gloom because as we know, traits can evolve. And so we wanted to know whether phytoplankton can evolve their traits and adapt to higher temperatures. So we did several experimental evolution studies. We conducted these studies with various diatoms, including, again, the model diatom Thalassiosira pseudonana. And so Danny O'Donnell conducted these experiments where he evolved different phytoplankton, Thalassiosira pseudonana in particular, for many, many generations and at 2 different temperatures, 16 degrees Celsius, shown here in blue, and 31 degrees Celsius. And after about 300 generations, we found that temperature optima adapted to higher temperatures. So these are the temperature optima of the high temperature evolved strains. And so basically, it looks like a lot of phytoplankton can adapt to rising temperatures, and a lot of different labs showed similar results. Basically, they found that phytoplankton can adapt. So, however, we should keep in mind that there are other stressors that may interfere with this adaptive potential. So Maria Aranguren-Garcís, a postdoc in the lab, set out to understand whether nutrient limitation may interact with adaptation to high temperature. And she conducted experimental evolution, adaptation to high temperature, under two different nitrogen levels, high nitrogen and low nitrogen. And what she found is that under low nitrogen, the adaptation to high temperature was not happening as easily as in high nitrogen treatments. So if you think that a lot of the ocean is nitrogen or phosphorus or iron limited, that may impede the evolutionary potential of phytoplankton in those parts of the ocean. So in addition to oceanic phytoplankton, we have a great interest in freshwater phytoplankton, and we look at traits of freshwater phytoplankton. And here's a short, brief example of some studies that we conducted on Lake Baikal. So Lake Baikal in Siberia is the world's oldest, deepest lake in the world, and it's undergoing a rapid warming. And so here's one of my, probably one of my favorite field photos. And if you can read Russian, it says, "I love plankton" on the t-shirt. So this is our crew of joint Russian-American team. And so I just wanted to highlight one result here. So Lake Baikal has a lot of endemic diatoms that bloom under the ice. And it turns out they're very sensitive to high temperatures. So these are the examples of thermal curves of some of these endemic diatoms. And you can see that these diatoms have positive growth rates only below, at temperatures below 12 degrees Celsius. So this rapid warming of Lake Baikal really will inhibit the growth of these endemic diatoms. And we have some models where we predict a gradual decline in the biomass of these endemic diatoms. So I see I'm almost out of time, but I wanted to highlight some of the exciting new directions that researchers around the world are pursuing, focusing on traits. And some of these directions are the following. So we are getting more and more information, and we are learning more and more about this interspecific and in particular intraspecific trait diversity. And intraspecific trait diversity is important because it provides us with a better understanding of the basis for evolutionary responses, because we need to have variation for evolution to act upon. And so there is an increasing focus on traits that can describe trophic interactions. And this is one of the exciting new directions. Then we're having a better idea of how traits vary along environmental gradients. And there are some really nice studies showing how traits of different trophic levels co-vary along various environmental gradients. Then we're learning more and more about trait evolution, and especially when we impose interacting drivers, such as high temperature and nutrient limitation, for example. And I'm particularly excited about the perspective of trait-based approaches when they're applied to various problems of societal importance. For example, we can use traits to predict community responses to climate change. And I'm really excited about using traits to understand and predict harmful algal blooms, both in freshwater and marine systems. And then we can use traits to help in ecosystem assessment and restoration. So, and I will conclude with some thoughts for future generation of limnologists and oceanographers that I think may be useful to keep in mind. So following advice of my PhD advisors, I think looking at the big picture and following your heart is really important in research. And focusing on the search for mechanisms can really inform our studies and understanding of ecological communities and ecosystems. And as we see that looking at other fields for ideas and approaches, both within the discipline of limnology and oceanography and across disciplines, can be really fruitful. And the final thought that I have is that don't be afraid to collaborate and actively search for collaborators who could complement your expertise. And don't be afraid to ask questions that you may think you don't have the expertise to answer, because there are always people who have that expertise. So, and if you build a team with complementary expertise, you really can tackle a lot of important fundamental questions. And with this, I will end my presentation. Thank you.
Thank you for that wonderful presentation. Let's wait a second and see if there are any questions that come into the chat. Well, not seeing any. I would like to thank you for the excellent presentation. Oh,
I didn't quite finish it, but it's just, I think it's an interesting component of looking at trait tipping points. And you showed this in your biodiversity work of sort of a temperature threshold. And I just wonder how pervasive are we exploring this notion of tipping points in terms of shifting communities, and particularly with these large scale changes going on?
Yeah, that's a great question. And you're absolutely right. In our work with Lake Baikal, we found that even gradual declines in ice cover and gradual increases in temperature can lead to abrupt shifts in community composition. For example, for some durations of ice cover, we found that the blooms of endemic diatoms completely disappeared. So I think these tipping points are widespread in ecological systems. So, and I think looking at what could be the indicators of those tipping points is an important area of research.
thank you. And then in Maria's work, negative N had slower or no adaptation. Was this scaled to generation time, or is there a smaller trait space for the cells to explore? And then secondly, does the potential mechanism give us some predictive capacity?
Mm-hmm. Yeah, good question. So what we found is that this interference of nutrient limitation with adaptation was probably due to higher nutrient requirements of high temperature adapted strains, clones. And so if they are limited by nutrients, they are not selected for, so they cannot, they're competitively inferior, even though they tolerate higher temperatures. So that was the mechanism. And so We scale it, we sort of took the generation time into account. So basically we ran those experiments with nitrogen limited strains for longer because they divided slower. So we wanted to have comparable number of generations. And so in terms of trait space, I don't know, we have to think about it, whether there is a smaller, probably potential trait space for those traits to evolve is shrunk because of this nutrient limitation. So it's a nice way to think about it, yeah. And predictive capacity, yeah, so if we can characterize nutrient requirements of different strains better, strains that differ in their tolerances of high temperatures, I think that would help us increase our predictive capabilities.